everybody to the fourth webinar of our educational series, CLL Care. Today we have with us Professor Silvia Di Aglio. She's a medical geneticist, full professor at the Department of Medical Sciences at the University of Turin in Italy. And Professor Di Aglio has held research positions at many prestigious institutions. She has received several awards. She serves on editorial boards of numerous scientific journals, and she's an active member of various professional organizations as well. And today, she's joining us to help us explore the world of chronic lymphocytic leukemia, and more particularly of Richter syndrome. So today's webinar is titled Richter Syndrome, Risk Factors and Molecular Insights. Professor Di Aglio, thank you very much for joining. The floor is yours.
Thank you, Anna, for the introduction, and good morning, everyone. As Anna said, I'm not a hematologist, and this is my disclosure to start with. But I'm not directly involved in patient care, but I've done research in the CLL field for almost 25 years now. So I just need to start by reminding you some of the house rules, if I can move it. Yeah. So you will be muted for the duration of the webinar, but you can always ask questions using the chat box. And if I, by any chance, don't see them, the Anna and her team will see them and alert me to them. So please ask any question you like, and I'll try my best to answer. So I think the objectives of this webinar today are threefold. First of all, I'd like to point the fingers to the key genetic factors that lead to genetic transformation, at least as we see today. To Richter's transformation is the transformation, as I will define you in a second, of a CLL into an aggressive lymphoma. The second point is to understand, to take this from the genetic to the molecular level and trying to understand the molecular mechanisms that underlie Richter syndrome, with the final aim of translating this knowledge into molecular insights to design novel therapies for this still incurable disease. So as I said, let me first start by telling you what we know about Richter syndrome. So the definition is that RS represents the development of an aggressive lymphoma in a patient that has a pre-existing CLL or SLL. The percentage in which this occurs in CLL patients is variable. It really depends on the kind of center. Center that see second and third referral patients generally have a higher percentage of Richter syndromes. People that centers that generally deal with untreated CLL see it in a minority of patients. Anyway, the percentages are, I would say, below 10%. The important thing in the definition is whether Richter's is clonally related or unrelated to the preceding CLL phase. And that means whether Richter's shares the same immunoglobulin B gene rearrangements of the preceding CLL phase. And so it's simple to determine that because it's just a test to check IgV gene usage. But this information is critical and should be obtained immediately because clonal relatedness is highly prognostic. In that if a patient develops a Richter syndrome that's unrelated to the preceding CLL phase, so he uses different IgV genes, then essentially the prognosis becomes that of a de novo diffuse large B-cell lymphoma. So it's far better than the prognosis of a clonally related Richter syndrome, which is incurable and shows rapid progression. And generally, the drug combinations that work for the LBCL will not work perfectly for clonally related RS syndrome. The genetic lesions that characterize RS, and I'll come back to this in a second, include deletions in the cyclin gene, CDKN2A and TP53. They include mutations in NOTCH1, and they include amplification of the very powerful oncogene MYC. So the first question that we may ask is, how does it develop? And the general answer to this question is that it develops in the context of high-risk CLL. Now, related to this is how do we define high-risk CLL? And I think that the concept of high-risk CLL has evolved significantly over time, so that in the beginning, back in the 80s and 90s, high-risk CLL was mainly a clinical definition. So that would be a patient developing a rapidly progressing, rapidly growing lymph node, for example. Then this changed abruptly at the turn of the century, which those of you who have been in the field long enough will know. The two seminal papers published by the Hamblin and group in the UK and by Nick Shirazzi in the US, where they showed that the mutational status of the IgG genes was prognostic, and that in fact unmutated IgG genes were defining higher risk CLL. And with that observation came the finding that CD38 expression could be a proxy for unmutated IgG genes, and so a marker for aggressive CLL. And then over the course of the following 10 years, a number of other markers, I've listed here just CD49D as an example of unfavorable prognosis were published. And so the definition shifted from clinical to, let's say, biological or a marker definition. And the following decade was the decade of the genetic studies. So people started using NGS to sequence to obtain maps of recurrently mutated genes, so that this, you can't read this, but here you would have a gene name and all these little dots represents individual patients. And so you see that this gene, for example, is recurrently mutated because you find it mutated in more than half of patients. And so these were the years when people defined all the genes that were recurrently mutated. And I would say that we have entered a decade of the omic data, which are not only genomic, but they're also transcriptomic, proteomic, and metabolomic. And then we will have, let's say, a comprehensive view of evolution of CLL over time. And I think the challenge now is to integrate all these layers. I took this image from a very nice review published by Kathy Wu and her team a couple of years ago, where she essentially says this, that the definition of high-risk CLL is a blend of clinical features and protein markers and genomic alterations, transcriptional alterations, and epigenomic alterations. And putting all these layers together, you really can see how we currently define high-risk CLL. So let's go back to Richter's. I told you that there are some defining genetic aspects that invariably happen during transformation. And this these were defined by seminal studies that were done about a decade ago by several different groups, Anna Shu in the UK and Riccardo Favara in New York. And others, which are summarized here. What you can see here are these bars here. They represent chromosomes, and you can see the chromosomes of CLL cells. You can see the chromosomes of so-called high-risk CLL, of Richter syndrome patients, and of diffuse large B-cell lymphoma patients taken as the control. And what you can see in blue are the copy number losses, so the deletions. And what you can see in red are the copy number gains, so the amplifications. And so what you can see at a bird's view is that CLL has relatively little blue and red, whereas DLBCL has a lot blue and red. And it's generally very different from everything else. So that sort of underlines the concept that Richter's and diffuse large B-cell lymphoma are separate disease entities. So it makes sense that they respond to different treatments and that they behave clinically different. The second point that I want to make is that in CLL, what you see is a dominant red band. This is chromosome 12, and that represents the subset of patients, which is about less than 20% in this cohort, that have trisomy 12. So they have 3 chromosome 12s, and you have a homogeneous increase of the color corresponding to chromosome 12. They also have a highly significant prevalence, 50, 60%, some people say more, of deletion 13q14 as a sole genetic genetic abnormality. And you can see here that this is a very small deletion on chromosome 13. And this is generally associated to a favorable prognosis. You can also see here chromosome 17, just a tiny bar corresponding to deletions where the TP53 gene is hosted, which is a minority of patients in unselected CLL. Then if you go down, if you scroll down and you look at high-risk CLL and Richter's, you can see that the complexity of genomic rearrangements increases. You can see that there's an increase which becomes the highest in trisomy 12 patients. So the trisomy 12 is, I would say, considered a marker of aggressive disease. And there's also a very marked increase, there's also a very marked increase in chromosome 17 deletions. So here you will see that patient in 3, a little bit more than a patient in 3 with Richter syndrome has 17p deletions. You can also see that there is the appearance of a deletion corresponding to chromosome 9, and this is where the genes coding for the cyclin CDKN2A and 2B are located. And this is highly prevalent in patients with Richter syndrome. And so you can see here that the genomic complexity increases with the increase of aggressiveness of the disease, maintaining a clear difference with Richter syndrome. Now, the second question linked to this is, how does the disease evolve? And I think you will certainly know that there are 2 models of cancer evolution. One is the linear model, where you have a number of genetic lesions. Here it's represented by a dot. This may be the minimum number of genetic lesions that define transformation. And then you can see that within this clone, there's a single cell that acquires one or multiple other genomic alterations, so that this clone emerging here will have all the genetic abnormalities of this cell plus some new ones. On the contrary, the branching idea of genetic evolution is that there are multiple different independent clones that do not necessarily share something with the primary, so that the emerging clone may be different from the founding clone. Well, when this question was asked, thinking about Richter's, the answer, and this was obtained by a study published about a decade ago by the Dalla Favara group, the answer is that it's mostly a linear evolution. And you can see it here, because if you look at genomic abnormalities, you will see that the majority are shared between CLL and RS. These are the abnormality in CLL only, and these are in RS only, and these are the shared ones. So you can see that in the majority of cases, abnormalities are shared, and Richter's has some more, which is consistent with the evolution, clinical evolution of the disease, going side by side with genetic evolution of the disease. You can see that in some instances, which are fewer in terms of number of patients, there are a branching model can also be hypothesized, because you have shared lesions. You have lesions that are typical of the most advanced phase of the disease, but you also have lesions that are typical only of CLL. And this is consistent again with a branching theory of evolution. So this is where we stand with genetics. Of course, there have been very seminal studies published by the Spanish group led by Elias Campo, and the Boston group led by Kadivu, where they have sequenced the exome and the genome of these patients, and they've come up with a list of recurrently mutated genes, and even genes that are not so recurrently mutated. But if we try to put them into pathways and make sense, so if we try to go from the genetic to the molecular, I think that there are 3 or 4 pathways that emerge so far. I would say that TP53 is significantly mutated. It is important, along with CDKN2A, in defining genomic instability and propensity to develop new genetic lesions. And then there are 2, let's say, oncogenic pathways, which are relying on Notch1, which is recurrently mutated in about 1 every 3 patients, and MYC amplifications that again define a proliferative attitude of the Richter cell. And in these past 5-6 years, several groups have invested in trying to go functional. So to take these observations into experimental models and to try and find what are the lesions required for transformation and what is their role in transformation. And I will just mention some of these, I think, seminal papers. This approach here that was published in a few blood papers 2-3 years ago was devised by the Dimitar Efremov group in Trieste. And what essentially they did is they used the cells from TCL1. You may know that TCL1 are mice that are transgenic for the TCL1 oncogene, and they develop late in life, so let's say after a year and a half, something like that, they develop a lymphoproliferation that somehow resembles a CLL. So they are considered a proxy of CLL. And the good thing is that you can actually take cells from TCL1 mice and use them to transplant in other animals. So what happened with Dimitar and his group was that they took these cells and they genomically edited them using the Cas9 approach, and they targeted a number of genes. So in those cells, they targeted the cyclins and they targeted TP53 alone or together. And then they put the cells, the genetically modified cells, they put them back into animals and they saw what happened. And what happened was that there was a positive selection of the edited cells. And that clearly showed that these mutations are oncogenic and that they do go well together. So that they then injected these cells, the control cells versus the genomically edited cells, back into animals. And what they observed is that in a much shorter time, the edited cells developed lymphoproliferation in the spleen, and there were many more neoplastic cells compared to the control. A similar approach was carried out always by Dimitar. The reference is missing here. It was another blood paper where they looked at the effect of toll-like receptors by editing IRAK4, which is critical in the TLR signaling system. And what they observed was that this was having a big effect only in cells that had an immune system. So that if you were injecting edited cells into NSG mice, which lack both TMB cells, so they lack an immune system, a functional immune system, there was no effect whatsoever. But if you were injecting these cells into immunocompetent mice, so into black 6 mice, for example, you would see an effect of the edited cells, which was due to, let's say, a microenvironmental circuit involving macrophages. In another approach published again in Blood 2-3 years ago by the German group, they could show a prominent role of AKT because they generated animals that had constitutive activation of AKT, again using the TCL1 model, and they showed that these animals had a very big spleen in a very short amount of time, indicating that these cells could proliferate. And what they could show was that this activation involved Notch1 signaling, because it was inducing expansion of CD4 T cells expressing a ligand. And so again, this author could show a microenvironmental crosstalk involving tumor cells and microenvironment using immunocompetent animals. Perhaps the newest approach in this was something published by the Kativu group, again, Elisa Tenaken, late last year, where she used a very elegant model to ask the question of what are the mutations that are really needed to induce Richter transformation. And the approach that they used, which was highly complex and very elegant, was to genetically edit cells in order to delete a number of genes. You can see them here, either 5 or 6 genes that they believed were essential. And so that once deleted, they would promote transformation of CLL into RS. And the idea here is to define the minimum number of genetic lesions. So for example, you can see the difference difference of when you have inserted 5 deletions or 6 deletions. So this is the 6 gene is TP53, and you can see here that you go from 11 mice having CLL and 8 having what you can define as an aggressive CLL, to 6 mice having CLL, 5 an aggressive form of CLL, and 6 having overt Richters. And if you do this instead of immunocompromised mice in immunocompetent mice, the number of animals that have RS increases even further, again underlying the role of the signals coming from the microenvironment. So I think that these studies, which are still need to be fully completed and fully elucidated, are going to give us an overview not only of the genes, but of the proteins and of the functions that are needed to induce transformation of CLL into Richters. Coming to a more practical question is aspect is, how do we treat something that is so complex? And I took this algorithm from a review published by Davide Rossi in Bellinzona, where essentially what he says, it's essential to confirm diagnosis. And you do diagnosis, you make diagnosis with imaging, PET-CT, and you can do a tailored biopsy. And then you can have different scenarios. You can have an aggressive CLL, which you will manage as CLL, or you can have a second cancer, which you will manage according to what cancer it is, or you can have something that's DLBCL Richter syndrome. So if you are here, the most important thing to do is, as I said at the beginning, to establish clonal relationship to the primary. Is it clonally related or clonally unrelated? If it's unrelated, you will manage that as a de novo DLBCL. And so you will make the molecular analysis that are important in distinguishing what kind of DLBCL you're dealing with, and then you will have a treatment algorithm. If it's clonally related, then essentially the only curative thing that we have at the moment is stem cell transplantation. So it depends really if the patient is fit to go into transplantation, and if it is fit, it depends on whether he or she has a donor or it is without donor. If it's unfit to go through transplantation, and it may be the majority of patients, because transformation may occur many years after diagnosis of CLL, so we may look at the relatively elderly population, it goes into clinical trials or follow-up. So there's no really established therapy regimen. And that means that there is a lot of investigation into understanding, into let's say defining drugs that can be used for these patients. So if you have to think what's there and to sort of list what's there and what's available, I think we can envisage naked antibodies, bispecific antibodies, or antibody drug conjugates in the antibody field. Then of course we may imagine CAR T-cell approaches. And what is perhaps more interesting also from a research point of view are novel agents, which may be used alone, or as I will tell you in a little bit, in combinations. So naked antibodies, immunomodulatory drugs, there's not a lot of experience and there's not a lot of success as far as I could find. There are some papers using PD-1. There are some experiences of PD-1 antibodies used in combination with ibrutinib, with some responses. In general, when used in monotherapy, it's an unsuccessful approach. There's some excitement about blinatumomab, which is a bispecific antibody. And as I'm sure you'll know, bispecific antibodies work by bringing, let's say, the immune system on target. So they have a moiety that binds CD3 and recruits T cells, and then they have a moiety that binds CD19, and that could cause, let's say, a T cell attack on the lymphoma. There's not much published, and there hasn't been a lot of progress in the past couple of years. So at the moment, it's just a list of a possible drug. CAR T cells have got two FDA-approved drugs, and they work by transfecting T cells with chimeric receptors that have, that's once they bind the target, they induce very potent activation of T cells, and then the T cells take care of their target. This may be certainly promising, and I think we'll hear a lot more about this in the next few years. Now, the other point are the let's say biological therapies, so therapies that go on target. And for to use those therapies, I think we need to have reliable models. And this for CLL has been a problem. For RS is an even bigger problem, because there are essentially, there is one cell line which has been published by a German group in Ulm. And that's about it. There are no other, it's very difficult to have growing primary cells. It's very difficult to have primary cells at all, because in most instances, they are lymph node biopsies. They either get fixed immediately, or they are very few in numbers. And so this has represented a major limitation that we were lucky to somewhat overcome by establishing patient-derived xenograft models. So some years ago, we started injecting primary Richter syndromes subcutaneously into immunocompromised mice. So the mice I was mentioning to you before, the LAC-TB and NK cells, they are essentially universal acceptors. So we got these fresh cells from patients, put them sub-Q and waited. In some instances, we waited many, many months, in some instances, even close to a year. And then at some point, something grew sub-Q. And we collected it and we started it, and we tried to see, first of all, whether the growth was reproducible. And in fact, it was, because here you have the 4 models. And when you inject them subcutaneously in animals, you can see that they have very different growth curves. So this green model grows very rapidly, and it will reach the size where you have to sacrifice the mouse in less than 3 weeks. Whereas this red model is much slower, and you will have to sacrifice the mouse in about 6 weeks or something like that. And the other 2 are intermediates. But the important thing is that they grow in a highly reproducible way, so that the green cells will always grow that aggressively, and the red ones the same. The second point is that they grow intravenously, so they diffuse. So when you inject them into the tail vein of these animals, you can see that they populate a number of organs, and you can find them in the spleen, in the bone marrow, and also somehow, some way to a lesser extent in parenchymatous organs. The important thing is that they retain a high degree of similarity with the primary samples. First of all, they do share the same IgV gene rearrangement. So here you have an example. This is the IgV gene rearrangement of this model, RS9737. This is the primary, and this is the PDX. It's identical, and also the percentage of somatic mutation is identical in all 4 models. And as you can see, we have 2 unmutated and 2 IgV mutated ones. Second, they are related to the primary because they maintain, in general, a high degree of overlap when you look at chromosomal abnormalities. So that you can see here that the ones that are deletions and amplifications, they are shared with their primary. And you can see that 3 out of 4, they have deletion of 17p and deletion of 17p and deletion of 17p. There is CDK N2A inactivation in this model. There's amplification of MYC. So they have the usual, I would say, reported chromosomal abnormalities, and these abnormalities are maintained in the primary and then in the PDX. So they seem to be a reliable model. They also maintain the same, or largely the same, set of mutations. You can see here that TP53 is also mutated, not only deleted, that there are mutations in NOTCH1. One has got the typical BTK mutation, and whenever you see the black dot inside here, it means that the mutation was already present in the primary tumor. You can see that the big drivers were present in the patient cells and were retained in the PDX tumor. What is also important is that if you go and look at gene expression profiling, so you move from the DNA to the messenger RNA level, and you look at globally what is their transcriptome, and you ask the software to make an unsupervised analysis, and that means to group your samples according to their similarity, independently of what they are, you will see that the PDX and the primary, they always go together, so that they are unsupervisedly grouped together in all 4 cases, so that the system recognizes that they are more similar between them than between the other samples. And lastly, which is also a critical point, their mutational landscape appears to be stable. So the way we work is we have kept these models going now for a few years, but we rederive them from the early passages every now and then, and the mutational landscape is stable. So these, for example, these cells, they carry this mutation in notch 1, which is there and remains there throughout the passages. They have all these set of mutations at the heterozygous state, and they stay at the heterozygous state. Some mutations, of course, are novel. For example, this one was not present in the primary, was acquired by the PDX, and then, of course, it wasn't. It stayed stable. What is relevant from a translational point of view is that they retain a high degree of similarity with the primary, and that they are stable, and lastly, that they grow IV. So because of these 3 things, we have extensively used these models in these years to test, let's say, functional hypotheses and to determine whether they could be used to test novel drugs. So what drugs can you test in PDX models? Remember that these mice do not have an immune system. So naked antibodies, bispecific antibodies, and CAR T cells are out of the picture. You can study antibody-drug conjugates depending on the drug conjugate, but if the drug targets something within the tumor cell, then they can be used in immunocompromised mouse. And of course, you can use them to study, let's say, small molecules that target, again, signaling pathways that are active in the tumor cell and that do not necessarily require interaction with the microenvironment. So let me just show you how we have used them. The first antibody-drug conjugates that we tested was a toxin, which is MMAE, conjugated to an antibody, which is ROR1. Now, those of you who are in the field of CLL may know ROR1, because ROR1 is expressed at high levels by CLL cells, and the naked antibody is used, has been used in clinical trials for CLL patients. And the interesting thing about ROR1 is that it's a neoantigen, so essentially there's no ROR1 expression in normal cells, so you wouldn't envisage much toxicity. So working together with a company that produced this drug conjugate, we tested it in our systems, and I will show you in a minute the results that we obtained. And also we have used this antibody, which is called CD37. CD37 is a B cell specific marker. So you will find it essentially on B cells, not necessarily only on tumoral B cells. So you will probably have as a side effect that also normal B cells are wiped out, but you shouldn't have toxicities beyond B cells. And this was conjugated to a second toxin. Let me just, which is amanitin. Let me just show you the results, for example, of a typical experiment. So when we inject the cells sub-Q, this is the engraftment time. You inject them, you let the tumor growth when it reaches 200 square millimeters, so a tiny nodules, then you treat. And we had different treatment schedules. For example, with the ROR1 ADC, we had 6 different treatments, 2 days, one from the other. And then after the end of treatment, the mice entered a watch and wait period. And there you had that animals that had received a vehicle, they started dying with the expected kinetics. This is the, let's say, longest model, and this is one of the short ones. And you can see here that the animals died, whereas the animals that were treated survived much, much longer. And there was a highly significant difference in the survival curves. Now, when we treated with CD37 ADC, the schedule was different because we had a single treatment. So there was engraftment again, just one single administration of the ADC, and then we waited. And again, you can see here, this is the mortality survival curve with this model, and this is the treated animals. And you can see that in this model, even a single treatment was highly impacted survival, which was less evident with other models. And we've never tried repeating the treatment, but this is something that may be worth doing. Now, the most interesting thing, perhaps also from the translational point of view, is using drugs that target pathways that are impacted during transformation. And one of these pathways is certainly apoptosis. We have looked at expression of the different pro and anti-apoptotic members, and we have done this together with a group in Padova. And as you can see here, for example, the prototype of the anti-apoptotic molecule BCL2, you can see that it's very heterogeneously expressed. In general, it is expressed at a much lower level in Richter's compared to CLL. And as you can see here in our 4 models, we had one which was completely negative for BCL2. And very interestingly, this model had sort of compensated loss of an anti-apoptotic with overexpression of MCL1 and BCL-XL. And so you can see that the profile of expression is different compared to CLL, but we could also show together with Antonella Rigo in Padova, that the priming was different of RS compared to CLL, and that the dependence of on anti-apoptotic molecules was different in Richter's compared to CLL. In general, if you use peptides that are BH3 mimetics, so that target these anti-apoptotic molecules, you will see that CLL cells, which are here, the dots, are maximally sensitive. They die, whereas RS cells, they show some degree of resistance. So they don't release as much cytochrome C, independently of the BH3 molecule that you have targeted. And in fact, here what you can see is that you have a decrease in a patient where we had cells from the CLL phase and the Richter's phase. You can see that there is an increase in responses to targeting all anti-apoptotic molecules. And here you see an anecdotal patient who had a CLL phase and then an RS-like phase, and then was treated and responded. And you can see here 1 week off, there was sort of a increase in aggressiveness of his condition, and this was paralleled by a decrease in apoptosis sensitivity. So that we came up with this, let's say, diagram where you have treatment-naive CLL cells, which are maximally dependent on BCL2. So for these cells, targeting BCL2 is a good idea. They will show maximal responses. As you can see, when they become relapse refractory, they are still dependent on BCL2, but they can show also dependencies on other anti-apoptotic molecules, such as, for example, as I will tell you in a second, MCL1. And then you can see here that Richter syndrome in general has decreased dependencies on anti-apoptotic proteins, particularly on BCL2. So it may be a good idea to co-target BCL2 with other agents to increase responses. And we have done this together with Andrea Iannello a few years ago by trying to co-target BCL2 and the other main anti-apoptotic molecule, which is MCL1. As you may know, BCL2 expression is relatively stable. The messenger RNA and the proteins are relatively stable, whereas MCL1 is a protein that's tightly regulated, so that it's dependent on continuous transcription, and it's also dependent on a protein degradation cycle, which is very active. So MCL1 gets ubiquitinated very rapidly and gets degraded. And so if you target the ubiquitination process, you can increase degradation of MCL1, and then perhaps you will take away an anti-apoptotic molecule. And what we showed was that in these cells, which are dependent on BCR signaling and which have a constitutively active BCR signaling, MCL1 degradation was impaired. I won't give you the molecular details because the paper has been published, but that was the starting point of thinking that if we could inhibit BCR signaling, perhaps by targeting the PI3K, and the PI3K is very active in our cells, and target BCL2, then we would have a dual targeting of two different anti-apoptotic molecules, MCL1 on the one side and BCL2 on the other one. And in fact, we showed this, and there was a paper that was published showing that there was a beneficial effect in using duvelisib, a PI3K inhibitor, together with a BCL2 inhibitor. And in fact, this is now in clinical trials. I won't show you the details of the in vitro and in vivo experiments that we did back then.
Professor, you have 5 minutes left, just so you know.
Yes. But I will just show you these curves. So here we had mice that were treated with a vehicle, a single PI3K inhibitor, BCL2 inhibitor, and the combination. And here you can see that the combination is far more effective than each inhibitor used alone. You can see 3 models, and you can see this 4th model where you have no effect. Why is that? Well, that is because that model does not express BCL2. So it's totally insensitive to BCL2 targeting. And that tells us that whenever we will think about treating a patient, molecular profiling is essential before starting treatment, so that the patient will have to undergo a genetic screen, but also probably a molecular profiling. And we did this in a cohort of Richter syndrome patients, and we confirmed that about 1 in 5 lacks BCL2. So this is not something strange happening in the mouse. I will go very fast here. There are different approaches to co-target MCL1 and BCL2. One we are pursuing right now is again to inhibit MCL1 transcription by targeting CDK9, and we are getting promising results. These are the results of the growth curves in animals. The other aspect that I won't go into details because of lack of time is metabolism. So this is also emerging as a, let's say, common pathway that distinguishes the CLL from RS. When you go from CLL to RS, there are a number of metabolic adaptation processes that occur, including an enhanced oxidative phosphorylation. And that makes these cells uniquely sensitive to metabolic targeting. There are some preliminary data which were obtained by the Spanish group, Ferran Nadeo, that was published in Nature Medicine a couple of years ago. We also show that these cells are sensitive to metabolic inhibitors. I will skip this part. It's again on targeting metabolism. Let me just finish with telling you the limitations of our current studies. I mentioned that at the beginning, we have difficulties in obtaining primary cells. We have too few cell lines, and we have mouse models that are not perfect. First of all, the mouse models that we use, the TCL1, well, they are highly artificial, and they will have a fixed set of genetic lesions. So they will have the microenvironment because we can use immune competent animals, but they will have a fixed set of genetic lesions. Mouse models like the PDXs that are more flexible because they are dependent on what a patient's had lack the microenvironment. And we know anybody that has worked in CLL knows that the microenvironment is essential in providing a number of accessory signals. So how can we overcome this? Well, 3D cultures, maybe. I've shown here something that was given to me by Cristina Schelzens at Raffaele. She's doing very highly advanced 3D cultures. They're still not at the, let's say, for everybody's lab, so because they require skills and they require 3D printers that are not commonly available. But let me finish with this. What I think is the main, let's say, conceptual current limitation that we are experiencing is that we go by educated guesses. When we translate that omic knowledge that has become available in terms of genomic landscape, proteomic landscape, etc., when we try to translate it into, let's say, applied medicine and into designing drugs, we go from an unbiased approach to an educated guess approach. And that may be a limitation that I'm sure the omic era will allow us to overcome.